experiment in the tropical Andes and show you a summary of some results with a focus on the most recent results on tree properties, tree leaf properties. So this experiment was started in 2008, so we already have 11 years now fertilizing tropical montane forests, and we are comparing different forest ecosystems at different elevations and how they respond to moderate N and P additions. The background is that the growth of tropical montane forests is assumed to be limited primarily by nutrients, by nitrogen and by phosphorus. So nitrogen should be more limiting in upper elevations, and phosphorus is supposed to be more limiting at lowland elevations. And you have already seen that the cycles in key elements have been severely altered by human activities. You remember the presentations by Wolfgang and also by Andre. And that is one more reason to study how these tropical montane forests will respond to addition of nitrogen and phosphorus. So our main hypotheses were that we, by the continued addition of nutrients, will have noticeable and persistent effects on different components and processes in the studied forests, and that these effects should differ between the compared elevation levels and between the type of nutrients added. So our study area, we are here in the south of Ecuador. We have three study sites from 3,000 meters close to Loja, here the Cajanuma entrance of the Podocarpus National Park, 2,000 meters the San Francisco Reserve, and 1,000 meters also in Podocarpus National Park. It's a highly diverse area. The Podocarpus National Park is famous for its high number of endemic species, not only in plants, but also especially in reptiles and birds. On the other hand, we have high deforestation rates in the area, and in the surrounding of the Podocarpus National Park, there's almost nothing left in the same size of intact forests. 3 elevations, 3 different forest types with different structure of forests. So we find here in the transition from lowland to montane forests still tall trees up to 40 meters, and then at the mid-elevation size, San Francisco forest at 2,000 meters with trees between 12 to 15 meters at the study site, and then smaller size trees at the upper elevation size. Our experiment, we started, as I mentioned, 2008. We are using 50 kilograms of nitrogen per year or 10 kilograms of phosphorus and the addition of both nutrients. So we have a replicated full factorial design at 3 elevations. We have 4 blocks per elevation. Each block contains all the 4 treatments. So a control and the 3 nutrient treatments. So I will give you a summary of the results from the first 10 years of our experiment. I will only shortly summarize the results from soil and below ground, because that was also a topic of some previous talks, and so I will focus more on tree growth and also on leaf properties, because the leaf properties were sampled last year. So these are recent results. So starting with soil and below ground. So the soil response was as expected. We find increasing availabilities of the added nutrients of phosphorus and also faster cycling of nitrogen where we added these nutrients. Then there was an increase of nitrate leaching, but only after nitrogen addition and not in the first years. That started after some years of continued nitrogen addition. If we add phosphorus at the same time, so the combined addition of phosphorus and nitrogen, there's no leaching so far of nitrogen. We had some very interesting results in mycorrhiza. There was a decrease in AMF species diversity and a directional change in composition. So we found that certain groups of the mycorrhizal fungi, they responded to phosphorus addition, became more common, whereas other groups responded more to nitrogen addition. The soil food webs, the microbial soil food webs were altered. There was a change in species composition where we added nutrients. Then in fine roots, we found that there were species-specific changes in the root architecture of common tree species. Oh, that was too fast. Is something missing? Oh, so I go on with tree growth, the second chapter of my results, and start with tree diameter growth. So we are using these diameter bands to monitor the tree diameter increment. We do that in about 2,000 individual trees. And our results so far show that we have an increase in growth after combined NNP addition at 2,000 and 3,000 meters. But not all species are growing faster after nutrient addition. So that was a bit complicated to separate the stand-level response from species-specific response. And that was why we started to use functional traits, so the properties of trees, to see if we can explain the growth response independent of species using the functional properties of trees. And what we did was we used the functional traits, and then we did an PCA, an ordination analysis, to see how the trees differ in their strategies. And each dot or each cross here represents one species of trees in San Francisco. And you see the red arrows, they represent the different functional traits. Here, for example, wood-specific gravity, which is characterizing slow-growing, long-living species. That was already mentioned by Matthäus. These species are normally reaching bigger sizes, are more resistant to mechanical stress, for example. On the other hand, nutrients, foliar nutrient specific leaf area, which is characterizing more acquisitive species, so species of faster growth and lower wood-specific gravity. One example is here, Podocarpus oleocarpus. That's one specifically conservative species. So with relatively high density in wood, high density in its vessels in the wood, then low contents of nutrients and a low specific leaf area. And what we found was that species like Podocarpus, or specifically Podocarpus, is responding negatively to nutrient addition. So where we add nitrogen, Podocarpus is growing slower than in the control plots, because in competition with the surrounding species, the surrounding trees, it's losing in competition. Yeah, but on the other hand, some other species, the more acquisitive species, they gained in competition and they grew faster. Then I come to the seedlings, because Wolfgang mentioned it in his talk. Adult trees, big trees, they are not very responsive. They stand like 100 years in the forest, and we don't expect a very fast response from trees. But we thought we should look at the regeneration also to get a faster response to our nutrient addition. So one point was what we found so far is that there was a reduced richness of species after nitrogen addition, only after nitrogen addition, not if we also add phosphorus. Then we had one experiment where we planted seedlings. These seedlings of Puteria torta at 1,000 meters. We produced seedlings from seeds collected in the forest, then planted them to the forest and harvested the seedlings after 1 year to see how they perform in the different treatments. And what we found that they were faster in growth where we added nutrients. But on the other hand, they also had higher herbivory and higher mortality rates. So that makes it difficult to see if the overall performance is better under nutrient addition or it's not so good. Then another experiment with seedling, we harvested the 2 most common species of seedlings at all elevations, so in total 6 species, and compared the different leaf traits, root traits from these seedlings. And we found that the response to nutrient addition was highly species-specific. So some species responded positively, other species did not. And there was no systematic effect of elevation. And now I come to the leaf properties. What we did recently, or that was in the last year, we collected leaves from the forest canopy, so from adult trees, but also from terrestrial herbs and from woody regeneration. And so this graph shows you data from the year 2018, so after 10 years of continued nutrient addition for the 3 different life forms. So this is trees, this is woody regeneration, and this is terrestrial herbs. And you see the foliar end contents comparing the 3 elevation. On the left, 1,000 meter, in the middle, 2,000, and on the right, 3,000 meter. And the different colors represent in white the control, in red, nitrogen, in gray, the combined addition of nitrogen and phosphorus, and in blue, phosphorus alone. So you see there were, in this case, in nitrogen, significant effects of nitrogen addition and combined NNP addition. Where we add these nutrients, we get higher concentrations in the leaves. And if we compare the different life forms, we see that there is a difference. So we have relatively low values of nitrogen in the canopy. Values are getting higher in the regeneration and in the terrestrial herbs. And with elevation, there is a decrease in trees and also a decrease in the other life forms. If we look at phosphorus, it's relatively similar. So again, we get a significant effect of the added nutrient. Where we add phosphorus, we find it then later in the leaves. But that is specifically strong, the effect in the woody regeneration and also in the terrestrial herbs. So they are really high, the values in phosphorus. And here the values, the nutrient concentrations are higher again in the understory compared to the canopy. Next parameter is leaf toughness. And you see here only effects of combined N and P addition and the understory in the 2 different life forms. And in this case, negative effect. So leaves become less tough where we add N and P. No effect in the canopy. In the canopy, there's an increase in toughness with elevation, and it's the same in both other life forms. So from these parameters, we thought that also might have an effect on herbivory. So the leaves became more attractive to herbivores. More nutrients, less tough leaves should be become more interesting to insects. And what you see on this graph is the leaf area loss in the different life forms. These lower panels show the same samples from 2018. The above panel shows herbivory measured by leaf litter traps. So we sampled the leaf litter from the canopy and used these leaves to quantify herbivory. And you see in the canopy litter there is a decrease in herbivory, so the rates are between 7 and 3% of area loss. In trees, values are a little bit, more or less on the same level in woody regeneration and a little bit lower in terrestrial herbs, but with a strong variation in herbs. So, but overall, there's no effect of fertilization on herbivory, so no increase, not the expected increase in herbivory. But what we also did some years before, we also looked at single species. We collected leaves from different species and compared the different treatments. And you can see here, for example, this upper panel shows 4 tree species from San Francisco, and the lower panel shows different life forms from San Francisco. Here's a terrestrial herb, a bamboo species, a fern, and a shrub species. And what you can see, there are some species which are affected, where herbivory is increased, especially where we add nitrogen, or the combination nitrogen and phosphorus. So I think that's one very important point. To summarize the leaf properties, in general, leaf properties are changing with elevation and differ between life forms. Nutrient concentration increase after addition of the respective nutrients. But in herbivory, there's no strong effect of nitrogen addition or phosphorus addition, only in single species. At the species level, we find a different effect. So it's the same like in tree growth that we find in leaf properties, only an effect on the species level. Species differ in their responses. Some conclusions. If nutrient effects change with elevation, There's no clear pattern of phosphorus limitation at lower elevation and no clear signs of N limitation at higher elevation. In most cases, the forest seems to be limited by both nutrients. Effect sizes changes with time. That depends. In some parameters, we find effects after 1 or 2 years and in others after 5 or 6 years. Very important on species level, trees' responses are species specific. And so in the future, probably nutrient deposition will change the structure and tree composition of Andean forests. So more conservative upper slope species will lose in competition and in abundance, and other species, acquisitive species, lower slope species, will gain and will become more common in these places. So that will most likely also have consequences for the resilience of these ecosystems against climate extremes, and also on the ecosystem functionings, the ecosystem services provided by these forests. I think my time is over. I have one additional slide, but it's only to summarize one let's say, visual summary of this. So that's I will be fast. That's the forest current conditions. So we have different species composition along environmental gradients. In this case, the soil fertility gradient with the turnover in species from poor soils to more fertile soils. And if we get increasing nutrient deposition, so we are changing the environmental conditions, and that's what we are simulating in NUMEX. And so currently we have many species like this Podocarpus I mentioned, or other endemic species. All these three are endemic to the area, and many of them restricted to the more poor soils. And we can expect that we get a shift in species composition, biotic interaction and function, and also species loss. So probably some of these species will not survive in the future under changed conditions. Thank you for your attention.
Okay, thank you, Jürgen. I don't know if Volker, your offer is standing that you give 5 of your minutes to him. Yeah, I can give. Yeah, then you can have some.
Only Volker.
I will make it.
3 beers, I guess, at least. Which is difficult currently, by the way.
No, I have some.
Okay, are there questions? Then you can have them back. Okay, the next talk is by Volker.
Thank you so much.
And he will talk about another long water consumption and cell diameter variation at different tree function types in area set in the north of
So, welcome everyone. First of all, I want Wolfgang and Jürgen stop bashing the old trees, because they also can react pretty fast, and I will try to show this. So, first of all, like a brief outline, first I will show some dendrometer data. I will show the data set. I will try to show that dendrometer data works as an indicator for vulnerability. Then I will show some set flow data, the data set, set flow data regarding modeling. I'll just do this for Matthias. And then I will also show some turgor and leaf water potential. And then I will show some take-home messages, because at the moment we don't have any conclusions yet. So first of all, you see a data set of dendrometer. At the moment we have currently 60 dendrometers applied below the towers and at the San Francisco plot 1. And we installed them in 2016, 17, and 19. Compared to the dendrometers Jürgen used, we have a resolution of 30 minutes. So they are also band dendrometer, but they automatically log the circumference. What I wanted to show you here is, hopefully this works, yes. I just want you to focus on those gray bars. They show the Verano del Niño dry phases, and there you can already see that the trees are not reacting that slow, actually, because we can see here some dry spells This is one of the bigger ones I will come to next. And usually during this time, the trees lose lots of circumference. So this is one of the loggers, perhaps you have seen them already mounted in the forest. So we're going a little bit into detail. Here you can see the tree circumference change for 3 weeks with 2 different short dry spells. You can see here and here, one with like 2.5 days of drought, dry spell, and 4 days. And what's important here is that all species reach the pre-drought values after like 1 to 2 days, so they are getting back on track. We will see the difference later. Important is also differences regarding the daily amplitude. If you see the green line, it's Weinmannia with the PFT of 10, and you can clearly see that it shows a very, very low daily amplitude, which somehow is not represented in PFT 9 of Podocarpus. So there we do not have a clear sign for the PFTs. Another thing is the lower growth rates, the higher the PFTs. You can see like Weinmannia and Podocarpus do not really grow compared to the other ones, Tapirira and Ocotea. So the thing I was mentioning before, here you can see the tree circumference change of 2 different species, Vismia and Tapirira, during the dry spell of 2016, which lasted 11 days. You can see this here in orange. Thank you very much. You can see that here in orange is the dry spell, and while Vismia was losing only like 0.7 millimeters, Tapirira was losing more or less 2 millimeters of circumference in just the 11 days. But this is not really the important part. The important part comes later, after the onset of the rain again, which is like indicated here with this dot. That for Vismia it was no problem at all. It just replenished the water of the stem loss, and like after several days it was already back at the pre-drought maximum. A total difference is seen at Tapirira. The pre-drought maximum was reached only after like more or less 2 months. So this could mean that Tapirira seems as a vulnerable species to like to even short droughts, and this also can lead to like a species dieback if we think about the future. So our different data set, now we're going to the sap flow. At the moment we're measuring 15 individual trees in total, also below the towers and at the San Francisco plot 1. We did the installation last year and this year, and there we have a resolution of 10 minutes, and we're putting like 3 pairs of Grenier sensors on each tree to get a mean value of it, and to make it kind of sure that it's not just an individual mistake of one side, which is not very good flown through. So here you can see 4 of the 15 individuals, and the sap flow values are in milliliters per square centimeters per minute. Usually you connect them with the sap flow area to get for the flow of water, like the complete water in the stem, but we want to compare them, so we don't do that here. And as you can see, like Jörg already had this page before, this graph, the PFTs respond again quite well to the percentage, the sap flow represents the PFTs quite well, with usually Tapirira, which is 7, then Ocotea, then Podocarpus, and then Weinmannia. If we go into detail here, we can see a clear differentiation into 2 groups, more or less, like the black ones, which are the lower PFTs, and the red ones, which are the higher PFTs. So this is also consistent with the shown lower growth rates of the higher PFTs. So just for Matthias, here we can show the VPD with the high synchronicity to the sap flow values. And yeah, as you can see, it more or less follows every little change in the sap flow. Perhaps it will help you or help us all in the future. So here you can see like a compared picture of the 3 weeks we are already talking about. So we have a high VPD, so lots of sunshine, the stem is depleting from water, and usually we lose circumference there. As a difference with the sap flow is pretty high during these times, and while the rain is coming back and the VPD gets lower, the tree is growing and like replenishing again. So then to the drivers of sap flow, like more or less the the leaves. We're also measuring turgor, and we're also measuring leaf water potential. Turgor we measure with SIM probes, so we have some kind of like a magnet procedure which we clamp onto the leaf, and it shows us like the relative turgor pressure compared to the magnetic pressure we put onto it. Here we have the leaf water potential measurements. Therefore, we use a Schollander pressure bomb, and we put like a leaf or a small branch into it, put pressure on it, and we investigate how much pressure we need until the sap flows out of it. Here you can see Hanthoanthus chrysanthus. That's, by the way, is the new name of Tabebuia glissanta, which Jörg was talking about earlier. And here you can see Centrilla montana. And there is, of course, as you can already, which is already visible, a very big difference between the two species. While the turgor of Hanthoanthus chrysanthus shows a very high amplitude, Centrilla montana does not show this, and this leads to the conclusion that there is kind of a different plant water regulation strategy. One is anisohydric and the other one is isohydric. But I don't want to go into detail with this one because we also have a poster of it. And as we said, like I could easily give like 5 minutes to Jürgen. I'm already coming to the take-home messages. And yeah, first of all, dendrometer data provides insights regarding vulnerability and reacts very fast, like to some things, and may lead to a possible species transition due to climate change and like the associated longer dry spells. At the moment, as every year, we're waiting for longer dry spells in San Francisco, but we hopefully get one this year. Then, as I already mentioned, sap flow and BPD are closely connected. There's a very good differentiation possible into two groups of the PFTs regarding the sap flow values, and that we have two different plant water regulation strategies with which we can easily distinguish anisohydric from isohydric plants. And that's already finished. Thank you for the attention. Thanks to everyone, and thanks to NCI.
Okay. Thank you for the concise talk. Obviously, it's not so much to say about the big trees. Okay. But you can ask more. We have plenty of time for questions. Ah, 3 questions, all simultaneously. So I just decide from left to right. So please start. I have yours.
So my question would be concerning the changes in abiotic factors that we have heard today in the lectures. What are the implications of forest diebacks if concerning your data, if you say that they are more stressed and getting probably closer to a point of no return? Do you have any implications about forest diebacks in montane?
At the moment, I think the data is not enough, but we're constantly waiting about more of these dry spells because that's more or less the only long dry spell we could at the moment investigate. We did some with all the dry spells from 4 to 11 days. We tried to investigate it, but it's only Tabebuia in the years in all these days of drought which reacts like very similar. So there are like 6 individuals reacting completely similar compared to all the others. And so I cannot really go into detail about forest dieback. It's kind of like far away, the forest dieback, this consideration.
Thank you for your response.
You showed on this one slide this comparison of 2 species showing different, like slide 5, like showing different reactions to droughts, like in Circumference. And you said the one species is more vulnerable to droughts because it takes longer to recover. But is it indeed that this plant is the second plant is suffering more by drought? Is it indeed showing like a lower performance or fitness, or is it just a different strategy?
Thanks very much. There's a problem in the dendrometer people. This part here is the very important part. I don't want to stretch it because it's not really important for us here, but as you can see, like the replenishment of water, usually we say that it's not allowed for a plant to grow if it's not water saturated. And we have the problem here that you can immediately see kind of a switch in the pattern here, like from a replenishment into a more or less usual growth pattern. And the problem is that there must be some, we think that there must be some internal destruction happening there, like destroying internals of Tapiria or having problems like with the refilling of vessels or whatever. So there should be kind of a problematic injury, I don't know. We're currently trying to figure out what's going on there. At the moment, we're taking microcores from those trees, and we're waiting for results considering cambial activity and like tree growth on a very high level of security that we can see the cambial activity or not. If there is growth or not, that's more or less the main question.
Falk, I was just fascinated of the fast reaction of sap flow on the VPD. Is there no delay? Is it completely in time?
Actually, as Erwin said, there's kind of a, it's still not really clear why the VPD and the sap flow is like so close connected. But yeah, the data says it is like that. So yeah, that's kind of impressive. Yeah.
Okay, are there more questions? One more.
Well, it's just a comment. Maybe it's related with the word of Mateus, and it's in this case that we are seeing how is the reaction with Tapirira. It's possible to model it in the future the community dynamics without this tree in a long term. It's like in a realistic scenario that I don't know. It's possible. Do you think it's interesting that it's
like if the climate change will happen here, like that there will be longer lasting droughts? It could be interesting, but I do not know if the model of Matthias will be able to. Could you implement something like this? Just giving questions back.
Yeah, I mean, what happens is that in that case, this particular combination of SLA and with specific gravity, which define the species, will not be there at all. So there would be like a restriction of the SLA range, which I will be throwing into the model. So that would be one example of an extinction of a species. So there would be a restriction in the range of possible trait values inside the area.
Yeah, because it would also be very interesting regarding Jürgen's talk about like
exactly.
Podocarpus and
yeah.
Okay, thank you very much. Then we move on. The next talk is by Diana Rojas, from seed dispersal networks to forest regeneration.
So it's a pleasure to us be here in Loja and in this symposium. I'm Diana Carolina Acosta Rojas, and with my colleague Maciek Barzik, we are going to talk today. We want to give you a guide, like a small tour through the entire cycle of plant regeneration with a particular information about what is happening in tropical mountain forest, and mainly in the forest that's surrounding us in Podocarpus National Park. So we are working with We are part of an institute in Frankfurt, it's Senckenberg Institute, and this work we are collaborating with Carlos Iván Espinosa here in UTPL and with Boris Tinoco for Universidad of Cuenca. So this talk is titled From Seed Dispersal Networks to Forest Regeneration. Well, just to start, I want that you imagine that you are in a forest with different plants of different ages. So you are with just seeds that they are germinating, and with seedlings, and with all trees. And then you start to ask or try to answer this question. Why do plants grow where they grow? And just to give you an idea, I have to say that plants rely on vectors to disperse or to move across the landscape. And we have some examples of these vectors, and they are animals, water, and wind. And when plants disperse, they may colonize open habitats to reach potential suitable microsites. They also may reduce exposure to competition, predation, and parasitism, contribute to gene flow within and between populations, and at the end, and it's related with our project, they can respond to climate fluctuations. So if we decide just to focus on seed dispersal by animals, it's interesting how to indeed frugivory and seed dispersal processes shape forest regeneration and community dynamics. So just to understand this cycle, we have this graph with at least the 4 main processes that are involved in this cycle, and you can see the seed removal, seed rain, seedling establishment, and fruit production. Then, how most of you know, in the previous project, we have some colleagues that they start with seed removal with the project from 2010 to 2013, development and validation of functional indicators for avian seed dispersal. And then, in this graph, we can see the wonderful dispersal network made by Marta Kitian and collaborators in 2016, when they found, it's amazing how 134 birds were feeding on 115 plants. So we have a huge network of more than 5,000 interactions. So as a result of this project, we note that plant abundance influenced that frugivore abundance influenced plant removal. So on the other hand, we have some other examples of the findings related to seed removal. And I have to say that elevation and forest fragmentation, they have an effect on plant frugivore networks also. You saw this graph in the morning, and we have in the upper part, and we have in the upper part 3 networks related with the continuous forests, and 1,000 meters in Bombuscaro, 2,000 meters in San Francisco, and 3,000 meters in Cajanuma. And here, the correspondent forest, fragmented forest in this case, and all the statistical parameters. So if we, as I told you at the beginning, if we want to give a tour in this cycle, now with all this knowledge about seed removal, we can start just to ask about what is happening with this seed rain. And then there is something that we want to do in this project. So for a better explanation, I just want to say that fleshy fruit production in the trees, that actually plants produce fleshy fruits, and then animals are feeding on these fleshy fruits, and they are just, when they are defecating or regurgitating the seeds, they are contributing to the amount of seeds that actually reach the soil. So this is that we call seed rain. And this seed rain is part of my PhD thesis because I want to ask this question: how do change in plant trait diversity translate into change in seed dispersal and seed rain along elevational gradients and in human modified habitat types? So in this slide, we have a part of our methods. Actually, in our talk, we don't have a lot of results yet because we need more time, and we hope in the future just to show you beautiful graphs as our colleagues. But then in this case, I just want to explain briefly that also we are working in 2 land use types, forests and pastures. We have three altitudes, and in every plot we have 18 traps, so we have an overall of 3,024 traps. This is a scheme of a plot in San Francisco, and then in these green points we are locating the traps. So as a small data about how is our results, we have a lot of morphotypes. We are spending a lot of time trying to identify and separate seeds and also seedlings. And in this case, we have a more or less, well, we have 100 morphotypes, and it's with more morphotype species in Bombuscado, which is the forest that is located in the lower elevation.
Hello. So as my part links to the next part of our plant regeneration cycle, I want to see you also and share with you about the results and ongoing experiment on the seedling establishment. Yes, so our questions regarding the seedling establishment, the experimental part, and the monitoring of natural processes, they involve 2 questions. One is, how do biotic and abiotic filters influence seedling establishment of plants with different size seeds? And the other one is, how do changes in the trait diversity of plant communities translate into seedling establishment and survival? To check the seedling establishment with the experimental parts looking for biotic and abiotic filters, we installed 18 subplots on each elevation, 9 pastures and 9 in the forest, which have area of 2 by 2 meters. So each treatment plot is 1 square meter plot, and we have 4 treatments. One is control, 3 treatments. So one is control, and then insecticide, fungicide, and both of them used every 2 weeks, just sprayed on the growing plants and the sown seeds. The ideal situation is to use 5 PFT species, because every PFT has one of species which we are going, or which we have sown. And going to our results, so far we have 2 species in 1,000 meter, which are Saurauia and Ogotea, and 4 species at San Francisco. These are Cecropia, Tapirira, Hieronima, and Clusia. The sowing experiment involves a lot of work, so also we need at least 3, in the ideal situation 5 meshes which protect seeds and then can be found in the field. So we need sometimes more than 1,000 seeds for entire experiment for 1 species. And as you can see, this is one of the subplots here in the pastures. And then after several months, in this case every 3 months, we check the seedling establishment, the height of seedlings, and leaf area, which results in leaf area loss due to herbivory. And also we measure leaf area index by the hemispheral photographies. And the same abiotic filters we will apply to the natural recruitment experiment, which is just a survey of the of the seedlings in all forest and pasture plots. So far we have
these results, which are 45 seedlings of 23 morphospecies in Bombuscaro, 61 seedlings of 23 morphospecies in San Francisco, and 39 seedlings of 25 morphospecies in Kahanuma. These are the results from 9 subplots of 1 meter square, which you can see here.
Subplots per plot.
Yes, which are per plot. So in total there are 27 per elevation. And here you can see some nice picture of Rubiaceae seedlings. And with Mateus we also started measuring light, like direct light measurements in all of the plots. So going from this side, when we do a seedling establishment, we also have to move to fruit production, which sometimes is the first part which we work on, and in this case we want to also link to the previous project. So the morphological trait space of fruiting plants you can see here, and that it's decreasing. Like the axes are the plant traits like height or seed size, and you can see that they decrease with elevation. As Marta also revealed, there is no much difference between fragmented and natural forest. The blue is natural forest and the red is fragmented forest. And you can also see the bigger dots are the species with interaction. So the bigger is the dot, the more interactions are. If the dots are very small, there are no interactions. And we want to investigate it better. So our question is, how does the diversity of functional traits in plant communities change with elevation and in different habitat types? And we measure specific traits of seeds and fruits. You can see, for example, Exclusia ducoides here. And the objective is to measure 37 species, which belong to all PFT species, PFT group, and I mean plant functional types. And so far, with at least one individuals, we have 18 of them, and which are completed, and have 5 individuals. We just have 7. Also, what we will do a part of that is the phenology. So from the last result, we have 3 months, which are May, June, and July, and you can see that phenology of trees in this month seems to be like the most, the higher in the fruiting trees percentage seems to be higher in Kahanuma. But as we use only PFT species for the phenology, it's not the entire view of the situation. And in these both projects, the previous one and our project, we try to get to know the entire plant regenerator project as a driver of vegetation dynamics. And our message for you is that understanding biotic and abiotic filters on plant regeneration is important for projecting regeneration dynamics, and observations and experiments help to identify future responses of plants to global change. Y para ustedes que hablan más en español, tenemos un mensaje que pueden llevar a la casa. Bueno, entonces entender los filtros bióticos y abióticos de la regeneración de las plantas es importante para pronosticar sus dinámicas de regeneración, y las observaciones y los experimentos de campo son herramientas fundamentales que ayudan a identificar las respuestas al cambio climático que las plantas pueden presentar a futuro. So, with the last slide, we want to thank all of people who work with us. We are very happy to work with you, and thank you for your contribution.
Thank you for the talk. We can have 1 or 2 short questions. Yes. I know you guys are in the early stages still of your dissertation and collecting data, but there's a lot of us here in Loja and in the UTPL who are interested in restoration, especially of the pastures. So I wonder if there's any observations that you've had that could help us in finding ways to economically, on large scale, restore degraded forests and pastures in the area.
Okay, so the part of our project is also to answer a bit this question. The problem is that we use only the species which we don't know before, so but they belong to PFTs, so also we have more or less idea how they work. And so far from the pastures, yeah, we can see that the bigger seeds, they have also, like in general, they have more more chance to survive, so the establishment is better. And I would say that in our project, we have less time to know if they can survive longer, but yes, they germinate in pastures. So this is only, the problem is that we have to protect with the fences the plants, because otherwise the cows, they will destroy everything. Anyway, they destroy sometimes, but as we protect it with the meshes and in the fences, it seems that they grow. So it's also interesting that there is no single plant in the natural regeneration plots, because the competition with grasses is so high that probably nothing will grow there. And in the plots which we firstly cut the grass and then sow them, like the seeds, they grow. So this is a good result, I think.
For my part, I have to say that for me it's also intriguing what is happening in the pastures, because with the traps, with the seed traps, well, we have some problems, but as far as I saw there, it's not possible to see at least fleshy fruit seeds in the seed traps. So if we think, for example, in this kind of regeneration, natural regeneration, that just to leave the pasture and then the birds, they can just arrive and then just disperse the seeds, seems to be difficult, but we need more time to understand what is the pattern. But for me it's interesting, because compared with other tropical forests that I have been, when you have traps and when you have perch, in our case we also have perch for birds, it's possible always just see some seeds from birds, but in our case it seems to be not possible, at least until this moment. So we know that we need more time just to know what is happening in pastures. Also, we are thinking to put other kind of traps in this site just to understand that. But it's challenging, and we hope that at the end of the project we can have some conclusions or recommendations about which species are just better to sow, or just some conditions, at least the minimum conditions that maybe we need to include in this kind of reforestation or regeneration process in pastures.
Okay. Thank you very much. We now have to move on. Come to the last talk, which is given by Nina Favik and is entitled Linking Traits of Tree Species with Herbivory in Tropical Mountain Rainforests.
Okay, so the last talk, but hopefully not the least. I have the pleasure to present this talk on behalf of Yves Tide, Samantha Solis-Oberg, and Roland Brandl, and we are working with the herbivores. So why do we work with herbivores? So insects, particularly herbivores, are the most diverse and also the most abundant animals in our terrestrial ecosystems, and they strongly influence not only the structure and communities, but also the functioning of our forests, for instance. And this work has shown that if you have herbivores in the system, you have a higher, without the herbivores, you have a lower NC and P flux to the soil. And if they are present, you have a higher NC and P flux to the soil, which means that you have a higher soil fertility and also plant growth, and there with carbon storage in the systems. However, it's not really understood how this works at the community level. As I said, that herbivores are so diverse, and Jörg mentioned the response effect framework already. We aim to use this framework to conceptualize the role of herbivores on herbivory in the system. So as a rough summary, the response effect framework first of all deals with the response to environmental changes. So we have a community, let's say, of trees or plants and herbivores there, and then we have environmental filtering. We have seen this already with Jürgen's talk, that if you add nitrogen or phosphorus, some species won't be able to compete there, so they will be filtered out, and in the end they will not persist in the system and will decrease in abundance or even be outcompeted there and not present anymore. So we have, at the end of this environmental filtering, a certain set or pool of species present. They are depicted in these different signs and colors, and then this set is kind of producing the feedback effect on the system, so is responsible for the functioning of the system. So, for instance, if you look at leaf traits, you have higher leaf area index, then you have these lighter or more palatable leaves that will be eaten by herbivores, most likely, and then you have less photosynthesis, and in the end, maybe less carbon storage in there. So it's important to understand how this trait diversity changes with climate change, where we use the space for time approach, or also from the forest system to the pastures. So our aim is to integrate these environmental changes on the response and effect graphs on the first trophic level, so the leaves, and then also look at the impact of both the environmental changes and the first trophic level on the herbivores, and maybe also feedback effects back on there, and how this all contributes to the herbivory in the system, and there was also how this maybe influence the model and the predictions in the future. So you have seen already, we also work jointly on these different sites from the lowland to Cajanuma, and we have these replicates, and we also try to work on these 52 different tree species in the functional types, both in the natural forest and in the pastures. You've seen this already. We have certain species, we have several species per PFT, and thus we end up with 12 to 20 species per plot on the lower levels and a little bit less, so 12 on the higher levels, where we try to sample at least for the herbivores. 4 replicates, 8 we won't manage, but it's still a tough job. And in our project, we have certain work packages. In the first work package, we assessed the plant palatability as a measure of maybe attractiveness to herbivores. So plants should invest, and that's what Jürgen already told you, more resources into defense in favorable environments, which means Bombuscaro. And so we predict that the palatability should increase with increasing elevation here. But it's not a only one-way option, but also there's feedbacks from the herbivores back to the plant species, so the herbivore communities should also influence the plant species composition and the functional traits there. So we assume that the palatability decreases with a CN ratio in the plants. To determine the palatability, we used feeding trials with the nymphs of the Jamaican field cricket. So we starved these little animals for 24 hours and then provided them with these leaf discs. And after another 24 hours, we measured, we measured the dry weight of these leaf discs to assess how much of the leaf material has been eaten by these generalist herbivores. And here are some first results. You see here the gradient from 1,000 to 3,000 meter, not all of the PFTs, because it's still, I think we're gonna look also at the species level more than on the PFT level, but according to our predictions, we see that the palatability increases with increasing elevation. So plants seem to invest less in secondary compounds in these harsh environments. In a second step, we used first traits of Jürgen measurements like specific leaf area and leaf toughness to relate it to these palatability experiments. And of course, these palatability experiments are to get a rough idea. It's kind of not the real world, what we see in the forest out there. This data we will get hopefully towards the end of the year. But what we see is quite astonishingly a decrease with increasing specific leaf area. You have seen in Jürgen's data that it should be the opposite way, so it's not the complete data, so maybe there won't be any results here or relationship here, and it's maybe also to the choice that we have only had this cricket species available for these experiments. And we didn't find interaction or relationship of palatability with the leaf toughness in our system. So I think having the final or real world herbivory data will provide us here with more insights on the relationships between herbivory and the plant traits, and particularly also if we have then the nitrogen content and all the other measures Jürgen is currently gathering. In the second work package, we are dealing with the herbivore communities and their traits. So I said already that herbivores can overcome plant defense by adapted traits. So we predict that herbivore biomass should decrease with successional stages, as these early successional stages have usually the thin leaves that are easy to digest by herbivores, while the later stages have rather thick leaves. And on the other hand, we also assume that herbivore communities are linked to plant communities, to certain species, that some are more palatable and others less, and thus we have priorities there, and we assume that they change with successional stages. How did we sample the herbivore communities? We went out, and currently another crew is present, and this one picture is apparently missing. So we go out there and fog the canopies of the trees. We have these lovely suits, so no one can identify us in the forest, and we use an biodegradable insecticide and spray for 10 minutes insecticide to the trees, and then we have these self-made traps below the canopy where we collect on 5 square meters all the insects and other invertebrates that come down. And then we have this lovely mixture of herbivores and of course also predators and other arthropods that are currently being sorted still. We identified, or we determined already, the biomass in the different feeding gills, and we plan to, because it's quite a lot of work and we have lots of student assistants working there, to determine also functional traits next year onwards. And we predict that trait diversity should decrease with elevation, but I think this will be, as currently we're doing the fogging in Bomboscaro right now, that will take another year until we have the data there. What we can say, and Jörg has shown this graph already, is that we didn't find a difference in herbivore dry biomass across the successional stages within our PFTs, and only for the Lepidopteran we find a slight decrease or a difference in the late successional stages towards the early and mid-successional stages. And it's all data only from 2,000 meters, I have to say. We then looked also at the composition of the herbivores, and what you can see here in this NNDS plot, early successional in red, the different species in different reddish tones. The same holds true for the intermediate successional in green and the late successional species in blue. And you see there's quite a big of overlap, so we don't find such a differentiation that we expected in the first place, but still you can see that apparently only some herbivores seem to feed on the late successional species. So I think if we come closer, on the one side, particularly across family level, on the herbivore side, maybe we see changes there that are not depicted on this high taxonomic level. And if we then plot this first axis of the NMDS ordination here against the early, intermediate, and late successional stages, you see there is no change, no significant difference, just a trend towards a difference on the late stage. But also we looked at phylogeny and family age, and we see that most of everything is explained at the family level, and that's due to we couldn't come across morphospecies towards the species level. So I think if we use the traits there, we will most likely see then maybe changes at this towards a later stage of the project. And in the last work package, we quantify the level of herbivory, so the leaf area loss, and we assume that herbivory and palatability usually are positively correlated, because that has been shown in the literature quite often, though we have only this one species where we could perform the feeding experiment with. And the second part of this work package is that we assume that the herbivore communities can influence plant composition and also the functional traits. That's what we aim to do at the end of this project, where we aim to integrate the abiotic and biotic factors and how they influence, they are directly or indirectly influences the herbivore process. But currently, the leaf area loss, the leaves are scanned, but we still have to determine, and a new person is starting to do this in November with the leaf area loss also related to different feeding guilds of the insects. And then we are currently using the same mesh traps you have seen already by Diana to determine the leaf area loss, as Jürgen has shown you before, on a whole year, a whole phenological cycle, so on a biweekly sampling in all the forest plots to see the leaf area loss on the community level, which will give us insights also how this is related maybe to abiotic factors and also to plant identity and traits. These two questions, I cannot present you any preliminary data, but we aim to provide this in the next year. And all of this data will then enable us to entangle the environmental changes, how the environmental changes affect the first and second trophic level, and then the process of herbivory, and these data will then be fed into the model and also in the response effect framework on the different processes to see how these biotic components influence water fluxes and carbon storage. And with this, I'm at the end of my talk, and I'm happy to answer any questions. Thank you.
Thank you, Nina. Yeah, we have time for some questions. Up to 2 hours, I guess, at 7 o'clock. No, only a few. Yeah?
Nina, I just want to know if you think that if there is an influence or an effect of these species that you are choose to make the experiment of palatability, because I understand that it's not a local species that maybe is not used to eat this type of leaves. Do you think that the selection of the species actually could have an influence or an effect on the results?
Of course, we aim to get the real-world data and kind of the leaf area loss of the species, where we have also kind of on the same individuals sampled specific leaf area, nitrogen content, and so on and so forth. But to use the local species for experiments is not possible. So you use generalist species, and in the first place, in our proposal, we aimed for 3 different generalistic species. Unfortunately, it was not possible to get them here in Ecuador. And usually Sphodoptera, for instance, caterpillar larvae, is used in many of these feeding experiments. That is not available here, but also like cricket species have been used very often in different parts of the world, where these feeding experiments were then later on related to, or were used to relate palatability measures to herbivory, and usually the relationship was pretty good. So using a generalistic species was the only option, because if you use a specialist, you won't assume or predict that they would eat everything, though apparently this cricket species did not eat that much of the leaves. So that's, but we will see in the end whether we still find a relationship or whether other factors contributed to this.
Okay, are there more questions? Does not seem to be the case. Then I conclude this afternoon session. Thank you very much again to all the presenters, and of course also to the auditory for the lively discussion. And have a nice evening and a nice joint dinner.